Hi, I'm Dr. Desiree Lucier, and welcome to my poster titled Evaluating Embedding Quality of Whole Brain Dynamic Parcellation Resting State fMRI on the CCNA and SIMON datasets. Resting state functional connectomics are useful for the identification of functional alterations in neurodegenerative disorders. Embeddings, representative of brain networks, are an important step in functional connectomics. It is currently unknown whether embeddings are able to represent different populations, such as different ages, sexes, or clinical populations, equally well, or their scalability for use with big data. Dypac is a Python package capable of generating embeddings, detecting stable whole brain dynamic parcellations in fMRI data by using a two-level clustering algorithm. Our aim is to evaluate the effects of age, sex, diagnosis, and scanner vendor on Dypac embedding quality using resting state functional MRI from the SIMON and CCNA datasets. The hypothesis is that the embedding quality will be consistent across these variables for both datasets. Neuroimaging data was pre-processed using the established fMRI pipelines for the Neuroimaging Analysis Toolkit, known as NEAC. Minimally pre-processed, unscrubbed, resampled data was used for the Dypac embedding models, which consisted of 72 scans from SIMON and 140 from the first release of CCNA after QC. Dypac uses a simple two-level clustering, one on sliding time windows and one on indicator functions of parcels aggregated over many windows. For the sliding windows, k-means runs directly on the time series, generating a family of parcels, which are represented with one hot encoders. The second-level aggregation clustering procedure gives the stable maps. Models were run with the MNI gray matter template and using 5mm smoothing, 20 clusters, and 500 states. The batch sizes were set to 2 for SIMON and 4 for CCNA to accommodate the different dataset sizes. Individual subject time series were used to generate R-square maps from the models to quantify embedding quality. For those R-square maps, a score of 1 is a perfect approximation and 0 is none, with a negative score being a very poor embedding. NISATS was used to compare R-square maps to evaluate for significant differences in embedding quality between sexes, age, clinical diagnoses, and scanner vendors, with FDR correction applied to each comparison. NILEARN plotting was used for visualization of the R-square mappings and contrast results. There were no associations of embedding quality with age and regression model for either data set. There were no differences in embedding quality between sexes or diagnoses for CCNA, or the comparisons between those groups that had 10 or more participants, such as AD, PD, SCI, and MCI, each with each other. However, the scanner vendor comparisons in the SIMON study revealed significant differences in 201 clusters for the Siemens versus Philips contrast, predominantly along the midline and temporal lobes, and 43 clusters for Siemens versus GE along the midline and outer cortex. But there were no differences seen for the Philips versus GE contrast. To summarize, Dypac embedding quality was consistent for age, sex, and diagnosis, and it scaled well for big data. Scanner differences were consistent with the literature suggesting systematic bias in embedding may result in site differences. This is an important step for validation of Dypac for dimension reduction in heterogeneous multi-site data. Future experiments will include comparisons with traditional ICA methods and investigate embedding component replicability between and within subjects and data sets. We believe that Dypac can increase the robustness and scalability of resting state fMRI as a biomarker, especially in the context of multi-site data acquisition. I would like to say a quick thank you to my mentor, Professor Pierre Bellet, also to Professor Aman Badwa, and the CIMXP Lab, and everyone for being here today. Thank you.